Hello and welcome to our discussion today about the structure and the function of the mitochondria. Mitochondria are many, mitochondrion is one. So the one unit is the mitochondrion, while so many mitochondrion would give us the mitochondria. So the plural form is mitochondria, while the singular form is mitochondrion. And it's fascinating how this mitochondrion is structured and the functions it plays. It's no longer a secret that the mitochondria are the powerhouse of the cell. They are responsible for the survival of the cell. But what is a secret is that it is also, the mitochondria is also responsible for the death of the cell. So it's an interesting paradox how the mitochondria provides the ATP that is needed, generates the ATP that is needed to power so many biochemical pathways and enzymes and cellular activities that are pivotal and very important for cellular survival. And at the same time, the mitochondria are responsible for the death of the cell, whether it's death by apoptosis, where we have programmed cell death, and the mitochondria initiates and activates pro-apoptotic pathways that lead to the cell death in a fashion that preserves cellular integrity and does not elicit inflammation in the nearby tissue, where the cell membrane is intact and is not fragmented, and intercellular lysosomes and these proteolytic enzymes do not leak out through the cell membrane and go to nearby tissue and destroy and damage that nearby tissue. And also when it comes to necrosis, and that is cell death by necrosis, it's a vicious way in which the cell dies by the fragmenting of the cell membrane, and there is a damage, there is a defect in the mitochondria because the mitochondria elicits apoptosis. And when there is, for example, hypoxia or loss of oxygen supply to the tissue, so the cells are no longer receiving the proper oxygen needed to power cellular activities, or that subliminal amount of oxygen needed to initiate apoptosis, the cell will have no time to process the activation of the apoptotic pathway, so the cell will collapse and damage and fall apart, and all of these intercellular lysosome, these proteolytic enzymes would leak out and damage nearby tissue. So we see that whether it's necrosis or apoptosis, we can trace back the source of cell death always to the mitochondria. Just remember, it's when it's cell death by apoptosis, it's initiated by the mitochondria, and if it's cell death by necrosis, it's because the mitochondria was defected, or there is not enough oxygen, or the mitochondria failed to provide the subliminal or the preliminary amount of ATP required to initiate the apoptotic pathway. So now let's talk about the structure and the function of the mitochondria. It's just so fascinating. I remember when I was a high school freshman. This was a very long time ago. This was in the year 2002. I remember I was 14 years old, and I was just so happy to start my high school career. And I remember my grandfather, God rest his soul, he insisted on taking me through and accompanying me in public transportation to the high school. I remember it was a specialized high school at the time. It was the one and only high school in my country, Sudan, and it was the high school that is everybody was trying to get into. It was the excellent, the top tier high school. And it was only one. And I remember when I gained admission, I was very happy. You have all of these just top, the best of the best were there, and I was just so happy to be part of it. And I still remember the first day of school, and we had this biology teacher. This biology teacher was so interesting in the way he used to teach us. I remember he used to tell us, do not draw except with a pencil. If you have a dot with a pen, you cannot even label your drawing or what you draw. This I remember the first lesson was the cell organelles or the cellular structure. And I remember he also talked about other organisms, other unicellular organisms. I think he talked about the hydra. Yeah, he talked about the hydra, and he talked about the promesium, and he talked about other prokaryotic or other unicellular creatures, and he also talked about the cell, the eukaryotic cell, and to show you the differences between these two different kinds of cells. And I remember he talked about each organelle in such a fascinating way, but when it came to the mitochondria, when it came to the mitochondrion, the way he taught it, it was just magnificent. I can still remember it more than 20 years ago. And when I reflect, my teacher at the time did not have the English proficiency to enable him to open the books of literature and these references to go and read all of this information that we know about the mitochondria involvement in apoptosis and mitochondrial implications in necrosis, and the all of these biochemical pathways, the citric acid cycle and oxidative phosphorylation, and how the mitochondria is responsible of all of these very important pivotal functions in the cell. I know he did not know about that, all about that. But what I do know is the way he taught it, he planted the seed. He just made us love biology, and he made us love, the mitochondria. So today, just talking about it just brought me back many years ago. So let's first, before we talk about the function of the mitochondria, we have to talk about the structure. So from the structure, we get the function. The knife is sharp. That what enables it to cut. So the function is cutting, but the structure is being sharp. The hammer is dull. So that hammer that is dull, what enables it to just hammer those nails, it's the property of being dull. Imagine using a knife to hammer down nails. Okay, you're not gonna have a lot of fun doing that. But let's talk about the structure. Please remember that scientists have always, hypothesized that the mitochondria evolved from ancestral prokaryotes that were engulfed by primitive eukaryote. So you have an ancestral prokaryote engulfed by primitive eukaryote. Ancestral prokaryote engulfed by primitive eukaryote. And what led them to that is the fact that the mitochondria mysteriously has circular DNA, which is a hallmark of the bacteria, prokaryotes. And the second reason is they realized when antibiotics, when microbicidal chemotherapy is applied, we have antibiotics applied to the cell, you would see that irrespective of which cell we're dealing with, the mitochondria is very susceptible and very vulnerable to damage by antibiotics. Just like who else? The bacteria. So these are the two key points that led scientists to investigate and hypothesize and investigate how the mitochondria evolved as ancestral prokaryote that was engulfed by primitive eukaryote. So we have two membranes, an outer membrane and an inner membrane, and between them we have an intermembrane space. The outer membrane is very permeable to solutes and ions, cations and anions and salts, but it's not that permeable to proteins. So if you take a sample from the cytosol and a sample from the content of the intermembrane space in the mitochondria, you would see that they have near identical content of their solute concentration, salt concentration, and also ions. However, the proteins, you will see that the protein composition in the cytosol is different than the protein composition in the intermembrane space, and that is because the outer membrane it does not have those channels, that are wide enough to permit the entry of these huge proteins. So the outer membrane has enough space to permit the entry of solutes and anions and cations and salts and so on. Okay, now let's talk about the inner membrane. The inner membrane is very tightly regulated, and the entry even of solutes and anions and cations is very tightly regulated. So you have this stone-like rigid membrane, the inner membrane, while the outer membrane is very permeable to solutes, anions, and salts. Okay. Now, this intermembrane space is very important for is very important for ATP generation, because there is an ATP rotor that generates. There's an ATP rotor that generates the synthesis of ATP. So we said we have an outer membrane, and then we have an inner membrane, and in between we have the intermembrane space where we have the synthesis and that ATP rotor that generates for us ATP. Okay, these foldings and evaginations are called the cristae. And they are very important to increase the surface area. So some mitochondria, if you remove this inner membrane and you spread out these evaginations to make them from the cristae form into a circular form, you will see that they are 5 or 10 times as big as the outer membrane. Okay, you need to know that this inner membrane has so many structural elements that participate in the generation of the ATP. Okay, then you have the core of the very inner core, and that is called the matrix. And in the matrix you have enzymes that are very important for the citric acid cycle. Very important for the citric acid cycle. Very important for the citric acid cycle. Also, the tricarboxylic acid cycle intermediates are also generated in the mitochondria. And what you should know also is that mitochondria is very important for metabolism. Like I said, the enzymes responsible for oxidative phosphorylation, which is the aerobic respiration of the cell, and the tricarboxylic acid cycle and the Krebs cycle and glycolysis take place in the mitochondria. However, in addition to metabolism, there is a new concept known as the Warburg Effect. The Warburg Effect, also known as the aerobic glycolysis, where glucose and glutamine and these metabolites are no longer used to generate ATP. They are used to generate skeletons, to synthesize lipids and proteins and macromolecules that the cell needs in order to survive. So yes, the ATP is very important to power many synthetic biochemical pathways in the cell. But the cell also, in addition to the ATP, needs these carbon skeletons that will cause the cell to shift, and instead of creating ATP from glucose, you would now have the cell shifting from anaerobic glycolysis into aerobic glycolysis, and now we will generate carbon skeletons from glucose. So now glucose will not be used to generate ATP as known in anaerobic glycolysis. There will be a shift known as the Warburg effect, and now the glucose will be used to create carbon skeleton for macro molecules such as lipids and proteins that the cell needs in order to survive. And you will see that the cancer cells have mastered this effect. The cancer cells do not waste all of the glucose that they have in order to generate ATP, but they make sure that they reserve some glucose and use using aerobic glycolysis to make the carbon skeleton needed for its macromolecules. Okay, so we talked about the lipid, protein, and building blocks used in the Warburg effect that takes place in the mitochondria. We know that the mitochondria is the powerhouse of the cell, and when there are apoptotic signals through the back complex, you would have pores into the mitochondria that will enable the leakage of cytochrome C and mitochondrial outer membrane permeability complexes. And then you would see that if there is defect or damage, there will be leakage there will be membrane permeability leading to the leakage of protons from the mitochondria, and that decreases the ATP production, leading to necrosis. And then we know that ischemia and injury of the toxins will lead to the defect of the mitochondria, leading to necrosis. And we know that the core matrix is where the citric acid cycle enzymes are located, and in the outer membrane you have the very permeable membrane, and the intermembrane respiratory chain proteins are found in the intermembrane space. I think that's about it. So thank you very much, and we'll see you to talk about this concept in more detail.